The overall goal of the sequential salt extraction procedure is to characterize protein binding profiles to bulk chromatin and evaluate how the binding is altered when the protein or its environment is manipulated.
This method measures the relative affinity of epigenetic regulators to chromatin and can be used to evaluate how those interactions change upon genetic, chemical, and environmental manipulation.
The main advantage of this technique is that it is a quick and inexpensive way to evaluate the binding profiles of chromatin-binding proteins.
Individuals new to the chromatin field often struggle with chromatin immunoprecipitation, or CHIP. Sequential salt extraction is an old technique that even inexperienced scientists can use to answer many of the same questions.
To begin the procedure, harvest 8 million cells from each cell line of interest. Wash each cell sample with 5 milliliters of ice-cold PBS twice. Resuspend each sample in 1 milliliter of a hypotonic buffer with protease inhibitors. Transfer the cell suspensions to 1.5 milliliter microcentrifuge tubes. Rotate the suspensions top over bottom for 10 minutes at 4 degrees Celsius. And then centrifuge the suspensions for 5 minutes at the same temperature at 6,000 Gs. Discard the supernatant. Equip a micropipette with a 1 milliliter tip. Add to the pellet 200 microliters of salt.